I'm an assistant scientist at the Cary Institute of Ecosystem Studies. Thanks for joining us for our weekly seminar series, sometimes in remote or sometimes in person, often in remote. You'll see a list of upcoming seminars listed here. So today we have Dr. Anna Trugman, who I'll invite in a moment. Next week, Dr. Regina Tulin from the University of Vermont will be joining us to talk about place-based education. And then the following week, we'll have Dr. Kyle Wickings from Cornell talking about microbial processes and managed grassland systems. So lots of exciting science in our near future. But today we have Dr. Anna Trugman, who I'll introduce in just a moment. A few logistics first, though. For those of you who are joining us from outside Cary, it'd be lovely if you could tell us where you're joining from in the chat. If you have any technical difficulties and want to get a hold of the folks who are organizing, please use the chat. If you have questions during Anna's talk, use the question and answer function, and I will moderate the Q&A at the end of her talk. So with all of that out of the way, I'd like to welcome Dr. Anna Trugman, our seminar speaker this week. Dr. Trugman's an assistant professor at the Department of Geography at UC Santa Barbara. She got her PhD at Princeton University in 2017 and spent a few years as a postdoctoral fellow at the University of Utah. Anna's a close collaborator of mine, and I've really enjoyed working with her. Anna has the unique ability to think across scales in ways that are pretty unique, and to integrate diverse data sets ranging from modeling to remote sensing to field work, and using those different data sets is able to kind of bring out pretty unique ecological insights. I'll also mention that she was just the winner of the 2021 Tansley Medal for Excellence in Plant Science, which is really exciting. And so I just want to take one more moment to say thank you for joining us, Anna, and we're looking forward to your talk.
Thank you so much, Winslow. That was a wonderful introduction. I'm going to share my screen now, and please, someone tell me if something happens to my video or audio verbally, because I won't be able to see you. But so I'm Anna Trugman. I'm hosted at the UCSB Department of Geography. I'm actually here in person. So feel free to talk to me after my talk if anything sparks you in the hallway or during lunch or happy hour. But today I want to talk to you about plant physiology, forest ecology, and carbon cycle uncertainty in a changing climate. And before I get started, I just want to acknowledge everyone in my lab group who's contributed either directly or through ideas to this work, as well as my collaborators and funding sources. And I want to start off today by giving you a brief overview of where I'm going to go. I'm going to talk about the motivation behind my research, and specifically forests in a water-limited world. I'm going to talk about using plant hydraulics to understand forest dry range edges and climate change responses, and this work was headed by my postdoc, Justin Mathias. And then I want to talk about the strengths and limitations of a physiologically based approach for predicting drought mortality in a changing climate. But first, forests in a water-limited world. So forests are this amazing resource. They host the vast majority of this planet's biodiversity. They also moderate surface energy fluxes, the hydrological cycle, and the terrestrial carbon cycle. And in my research program, we're actually really interested in their role in the terrestrial carbon cycle. And the reason we're so interested is forests are this really critical carbon sink. So this is a picture of the global carbon cycle over time. And on the positive part of the axis, we have net emissions due to fossil fuel emissions and land use change. And the carbon from these emissions can go into actually three main reservoirs in the Earth system. They can go into the ocean. And if you look, the amount of carbon going into the ocean doesn't actually change a lot on short time scales. This carbon can go into the atmosphere. And this is what I think of when I think of anthropogenic climate change, is rapidly rising atmospheric CO2. But if you actually look at the amount going into the atmosphere from year to year, it's highly variable. And the reason it's so variable are terrestrial ecosystems and plants. And so currently, terrestrial ecosystems are sequestering about 30% of anthropogenic emissions annually. So they're doing this huge service in terms of mitigating climate change. But if you look back on the record, they're really sensitive to changes in water availability. And so looking forward into the future, where we expect increased CO2, which can increase water use efficiency and productivity, there's a lot of uncertainty how terrestrial ecosystems will respond, because on the one hand, we have this physiological benefit, but on the other hand, the radiative effect of CO2 can increase instances of drought, heat, and fire-induced mortality. This is a major drought mortality event that occurred in the past decade in California. And these types of mortality events not only have drastic effects for biodiversity, but they can also release a large amount of carbon back into the atmosphere within a relatively short time period and cause a positive feedback on climate change. So really understanding where and when we expect to see a benefit from CO2 fertilization versus a net negative due to these radiative effects of CO2 in the atmosphere is of critical importance for understanding how to mitigate climate change and adapt, and also how to preserve our forest systems that we love so much. And drought mortality is actually already a global problem. So this is a map of major drought and heat-induced mortality events around the globe over the past several decades. And each one of these points is a major mortality event that has the potential to release this carbon back into the atmosphere and cause a positive feedback on climate change. And there's actually potential for it to get substantially worse. So with climate change, we expect changes in precipitation. And these are fairly uncertain, but also as we warm the atmosphere, we increase the amount of moisture the atmosphere can hold. And this puts additional atmospheric water stress on trees. And actually, water availability is really important even from an Earth system view, and it strongly influences the land carbon sink. So this was a very interesting study performed by Humphrey and colleagues and published in Nature. And they actually looked at the detrended atmospheric CO2 growth rate, which is shown on the left-hand side of the x-axis in black. And actually the atmospheric CO2 growth rate decreases increases as you decrease along the y-axis. And then they compared it to terrestrial water storage as measured from the GRACE satellite. And this is, you can see on the right-hand side of the y-axis as increasing in blue. And years with anomalously high atmospheric CO2 growth rate corresponded with years that were anomalously low in terms of terrestrial water storage. And actually the converse was true as well. Years that had anomalously low atmospheric CO2 growth rates had anomalously high terrestrial water storage. And so with that motivation, I want to talk to you first about using plant hydraulics to understand forest dry range edges and climate change responses. So we might better understand how forest ecosystems might respond to these anticipated changes in climate over the upcoming century. And this work was headed by my postdoc, Justin Mathias. And in particular, we wanted to answer 4 key questions. How does plant carbon balance or carbon use efficiency, which we defined as net primary productivity over gross primary productivity, so a metric for how much of the carbon that plants gain through photosynthesis is lost to respiration. How does this vary across space with climate? Are declines in carbon use efficiency prognostic of forest shrubland transitions at dry range edges? How will projected changes in climate impact carbon use efficiency and forest biogeographic extents? And finally, what are some major physiological uncertainties in understanding forest range extents through this framework? And so to answer this question, our primary tool was a plant model. It's an optimization-based model that includes a realistic representation of gas exchange, a detailed representation of plant hydraulics. And in the model, photosynthesis is a function of plant physiology and climate. So the climate that's used to force this model includes soil moisture, atmospheric vapor pressure deficit, CO2, and temperature. And the model predicts how environmental conditions impact plant hydraulic function and carbon gain. And so now I want to talk about some of the experiments. First, we validated this simple plant hydraulics model with observations of carbon use efficiency globally. And that's what's shown here in the figure on the right-hand side are different carbon use efficiency values that were compiled by Ecology and Prentice around the globe. And redder values indicate higher carbon use efficiency, so less of gross primary productivity being lost to respiration. And we also validated the model with forest biogeographic distributions using a LiDAR-based data set of canopy height distributions globally. And we were actually pretty satisfied with the model performance and felt like we could use it as a tool for understanding future patterns across the globe in terms of forest extent and carbon use efficiency. And so we performed a set of experiments where we forced the model with both historical, so 1975 to 2005, and future 2080 to 2100 climate conditions to look at how carbon use efficiency and forest distributions changed with those changes in climate. And we actually varied the respiration sensitivity to temperature according to 6 widely used equations that are incorporated in a variety of land surface models, but aren't generally systematically compared, so that we could understand how our understanding of respiration via these empirical functions impacted model predictions. And one really exciting result that Justin found was that model simulated carbon balance was actually a pretty useful tool in capturing forest shrubland transitions at dry range edges. And that's what's shown here in this figure, but it's a little complicated, so bear with me as I walk you through piece by piece. So on the y-axis, I show model simulated carbon use efficiency or net primary productivity over gross primary productivity. And that's based on the carbon use efficiency due to a grid cell specific climate across the globe. On the x-axis, I show canopy height from this LiDAR-based data set in meters. And each one of these different points represents a different grid cell across the globe. And yellower colors represent a higher density of points in this distribution. These dashed lines represent the interquartile range of observed carbon use efficiency values globally. And this distribution is actually bimodal. So there's one portion of the distribution where carbon use efficiency, as predicted by the model, is independent of canopy height. And that actually corresponds to higher latitude systems that generally have a higher carbon use efficiency as indicated by these greener to yellower values on the map due to lower temperatures, so you have lower respiration. And the model is actually not great at capturing transitions to shrublands at higher latitudes because the cold temperature limit processes are not incorporated in this model. But if we look at this other portion of the distribution, where we generally have lower carbon use efficiency values as indicated by bluer or purpler colors, this corresponds to tropical and semi-arid systems across the globe. that due to warmer temperatures have an overall lower carbon use efficiency. The model's really able to capture this transition from forest to shrublands through a sharp decrease in carbon use efficiency that you can see below this about 10 meters in canopy height. And that's really the model anticipating that from the climate conditions, plants are not able to, the respiration costs are so high that they're not able to exist long-term in those climates due to a negative carbon balance. And so we then wanted to really understand how future climate conditions might impact forest carbon use efficiency and dry range edges. And so what we show here is the difference in carbon use efficiency, average global carbon use efficiency between future minus historical observations. And each one of these points represents a different global simulation. And these different colors represent simulations with different respiration functions used to calculate total plant respiration and then carbon use efficiency. And this top panel here shows the total predicted change in carbon use efficiency in future minus historical. And you can see that overall there's a predicted net increase in carbon use efficiency, but it can vary depending on the different respiration function used. And this is mainly due to a CO2 fertilization effect. So Justin was able to run different factorial simulations where he systematically manipulated each of the climate forcing factors. And higher temperatures had a moderating effect to CO2 fertilization, as did higher vapor pressure deficits. And overall soil moisture didn't account for much of this change. But these globally averaged values don't tell us the whole picture. And so Justin actually looked at a more spatially explicit, for a more spatially explicit understanding of what was going on. And he actually found that although carbon use efficiency increased in core forested areas, there was a net decrease in total forested area, so a contraction of forests globally due to decreases in carbon use efficiency at dry range edges. And that's what we try and illustrate here with these different, using a biome map across the globe with these different biomes, including tropical grasslands, tropical conifer, temperate conifer, tropical moist broadleaf, desert, temperate grassland, montane grassland, temperate broadleaf, Mediterranean, and boreal forests. And so what's plotted here is the response ratio, which we defined as the increase in forest area or forest encroachment into a particular area, divided by the log of the increase divided by the decrease in that forested area. And so positive parts of this response ratio indicate increasing forested area in a particular location or forest encroachment, and negative values correspond to decreasing forested area. And overall, we found that there is potential for forest encroachment in tropical grasslands and forest expansion in tropical conifer forests. There were several different biomes where there was no clear net change. And we found potential for substantial decreases in areas classified as montane grassland, temperate broadleaf forests, Mediterranean, and also boreal forests. And for example, in the North American boreal forests, there's already evidence for substantial declines in forest productivity and resilience at drier range edges, dating back to some papers in early 2000s related to declines in white spruce growth. And what's actually really interesting is that though you didn't see at the global average this different respiration functions mattering in a huge way, the respiration function choice does matter, particularly at higher temperatures that are climatologically relevant under some climate change scenarios. And we have really little data to fit these empirical functions at those temperatures, which explains potentially this divergence. And so Justin actually did a breakpoint analysis and found that carbon use efficiency between the extreme members of these distributions really diverged starting at about 36.5 degrees C. And just for context, if we're looking along the steepest emission scenarios, there are some forested regions where the average temperature is above this 36.5 degrees C. And if we look at the maximum growing season temperature, there are broad swaths of forest across all forested continents that exceed this temperature. So it's an increasingly important unknown as we move forward into the 21st century. So just to summarize, I showed in section 1 that declines in plant carbon use efficiency can explain canopy transitions from forest to shrublands at dry but not cold range edges. Under climate change scenarios, carbon use efficiency may increase in core forested regions, mainly due to the CO2 fertilization effect, but decrease at dry range edges due to water limitation and high temperature, corresponding with a loss in total forested area. And importantly, this estimate actually doesn't include a lot of disturbance drivers and anthropogenic modifications of forest range edges. So it's a really conservative estimate of the processes that might cause contractions of forests. And finally, the sensitivity of respiration to temperature is an increasingly important unknown with the anthropogenic climate change. And so now I want to talk about, and I hope I convinced you from Justin's analysis that physiology does have a lot of merit in understanding broad geographic patterns in forest sensitivity to climate. But I want to talk about some of the limitations associated with solely physiologically based approach, particularly when we're trying to predict drought mortality in a changing climate. And in particular, I'm going to ask several key questions. Can drought-driven mortality be predicted from an understanding of plant physiology and plant hydraulics? What are the requirements and spatial scope? And what are possible missing mechanisms stymieing mortality predictions? And I want to illustrate the strengths and limitations of physiology, first by contrasting two different experiments with the same plant hydraulics model, the Sperry model. And actually both of these studies were headed by Martin Venturis. And the first study is a local controlled aspen sapling experiment performed at the University of Utah Research Garden. And the second is comparing the same model parameterized with species-specific traits to observed mortality in the forest inventory data across the Western US. I want to talk a little bit about the model first, so it's not as much of a black box. The model is a stand-based model with individual trees. Plant physiological traits include leaf area to basal area, maximum plant conductance, xylem vulnerability curves for roots, stems, and leaves, and maximum carboxylation rate. The schematic on the right actually kind of gives you a sense for what the input variables are and the plant physiological traits. Meteorological forcing variables include hourly shortwave radiation, air temperature, precipitation, wind speed, and atmospheric vapor pressure deficit. And outputs include a number of really useful diagnostics for understanding both plant function and plant sensitivity to the environment. So things like radiation, net carbon assimilation, plant water potentials, which give us an idea of plant water status, and percent loss in hydraulic conductivity. And I want to pause here to give a little bit of a physiological 101 lesson so that we can interpret particularly the physiological diagnostics related to plant water status. And so as you increase either dryness in soil water potential or atmospheric dryness, this puts additional stress on a plant's xylem or stem, and the internal xylem sap pressure becomes increasingly negative as you decrease soil water potential or increased atmospheric vapor pressure deficit. And that's what's shown on the x-axis here is xylem sap pressure becoming increasingly negative as you increase along the x-axis. And as this happens, the plant loses conductivity, which is shown on the y-axis. And you can see there's this really nice vulnerability curve describing how the plant loses conductivity as a function of xylem sap pressure. And importantly, different trees have these different percent loss in conductivity curves, and this corresponds to trees that are either more resistant to water stress. And so in this case, this blue juniper has less of a percent loss of conductivity at more negative xylem sap pressures, so it's more resistant to drought stress, and plants that are more vulnerable to drought stress. And actually, what's really nice for us modelers is that physiologists have identified thresholds that are roughly between 80 and 90% in terms of percent loss in conductivity that correspond to lethal levels of stress. So if we know what the water availability is and we have an understanding of plant hydraulics, we can anticipate if trees have experienced a lethal level of stress from this percent loss in conductivity, which is conveniently an output of this Sperry hydraulics model. And so for this local controlled aspen sapling experiment, what was really nice is we had the luxury of measuring plant-specific physiological traits. And so usually you might have species-specific averages, but in this case, Martin Venturis went out and measured plant-specific traits in his study. There was local tower meteorology associated with the University of Utah research garden that we could use to force the model. And these different saplings were planted with 3 different drought intensities as well as a control. And over the course of the growing season, Martin was able to observe plant status and then force the model with the same climate those plants were seeing and look to see what the model predicted outcome was and compare it to what we actually saw in these different saplings. And in particular, Martin compared model predicted hydraulic stress or percent loss in conductivity to actual sapling mortality. And this shows the model simulated whole plant loss hydraulic conductance, and then compared to the aspen mortality that we saw in the research garden. And on the x-axis is the hourly time step into the growing season. On the y-axis is the percent loss hydraulic conductance. And these 4 different panels correspond to different drought treatments. The dashed lines at the top in either pink or red represent this roughly lethal level of hydraulic stress as predicted by the model. And the black or red wiggly lines are the model predicted percent loss hydraulic conductance. And when the lines transition from black to red, that corresponds to when the tree died in the research garden. So Martin was able to do a really rigorous comparison. And in these bottom 2 panels, in the drought recovery and the moderate drought experiment, the seedling survived in both cases, and the model actually didn't predict a lethal level of stress. In contrast, in the top 2 panels, where there was a severe drought experiment, the model predicted at different points in the growing season that the sapling experienced a lethal level of stress. And actually, we observed sapling mortality in the research garden. It didn't correspond to exactly the time when the model predicted this lethal level of stress occurred, but overall, we were actually really heartened by these predictions, and we felt like the model was doing quite a good job capturing this lethal stress from physiological principles alone. And so then Martin really wanted to extend this study to see if the same plant hydraulics model was able to predict drought mortality at larger spatial scales. And so in this study, we selected 8 tree species that grow in monospecific stands across the Western US. And the reason we did this was just for tractability of the study, because we had to parameterize each of these species with literature reported species traits and initialize the demography for the model. We used meteorology from a gridded data set called the Princeton Global Forcing data set. And we performed a 5-year model spin-up to spin up the soil moisture, and then a 10-year simulation corresponding to the mean period between forest inventory remeasurements in the Western US. And we tested to see if model predicted percent loss of whole plant hydraulic conductance was prognostic of stand mortality during this 10-year period. And this shows the 8 different species that we examined, including aspen, Colorado pinyon, Douglas fir, lodgepole pine, one-seed juniper, ponderosa, single-leaf pinyon, and Utah juniper. And each one of these either black or red points represents the different plots. Black plots correspond to plots where there was no mortality, and red plots correspond to plots where there was observed mortality. And this shows the percent basal area mortality on the y-axis for each of these 8 different species, and versus on the x-axis modeled maximum percent loss conductance during this 10-year period. And yellower colors indicate a higher density of points here. And overall, we were actually really disappointed with the model performance. In a lot of cases, the model substantially over-predicted stress, and we didn't really see any mortality in the inventory. And so there's some clear scaling problem between the and gap between when we go from this controlled experiment in the research garden to trying to predict drought mortality at broad spatial scales. And actually, there haven't been a lot of studies that have tried to do this. One of the other few studies that have tried at a regional scale was performed by a different Martin Decaw, using the cable land surface model. And actually, the model structure is substantially different from this Sperry model. It's a modified big leaf model, but it does have a pretty similar version of the Sperry plant hydraulic scheme. And Martin Decaw and colleagues applied this model to the millennium and big dry droughts in Australia. And this shows model predicted stress or percent loss in conductivity, which is that same metric that Martin Venturis was using. And actually, they didn't have detailed inventory, so they used NDVI and vegetation optical depths as proxies for mortality. And so on these panels, browner colors correspond to areas potentially suffering from more mortality. And you can see the model doesn't predict a lot of high-level stress in the coastal areas where there's observed browning during these droughts. So there's really kind of an underprediction of tree stress in this context relative to what we think we see from the remote sensing data. And so I want to talk now about what missing mechanisms might explain why tree drought mortality is so hard for us to predict from our physiologically based perspective. And so one factor is that our understanding of plant available water is really limited, and actually where the plants are getting water from and how it varies across fine spatial scales and over time is really hard to understand. And so, for example, in a relatively recent study, Erica McCormick and colleagues showed that across pretty broad spatial areas, plants are tapping bedrock water. And so green corresponds to plants tapping bedrock water every year between 2003 and 2017. And purple corresponds to years where they're tapping bedrock water some year, and the grayed out areas are areas where they weren't able to determine based on their method. But this subsurface picture is actually really important in understanding the variations we're seeing across the landscape. And it seems somewhat intractable because it's hard to measure the subsurface, but people have already been doing some really innovative studies using above ground physiological diagnostics combined with process-based models to basically back out what we think the roots are doing. And so this is one study by Scott McKay and colleagues, but there have been a few more studies related to this. And I really think this is an important avenue of research moving forward. The other possible missing mechanism is additional mechanisms related to physiological damage and recovery. And so you can actually look at trees that end up dying during or post-drought event. And this shows some trees in Colorado that dying trees are shown in red and surviving trees are shown in blue post drought in the early 2000s. And you actually find that they the dying trees are able to maintain normal ring widths for over half a decade before ultimately suffering mortality. And you can actually explain this pattern using a simple physiologically based tree model with different levels of xylem damage, where lethal levels of xylem damage, the tree is able to grow at normal ring widths in an attempt to repair its xylem, but ultimately dies of carbon starvation after several years. And so potentially incorporating these damage and recovery mechanisms might be able to help us anticipate these patterns across the landscape more skillfully in a model context. And then disturbance processes such as biotic agents have a lot of potential for complicating our mortality predictions. And I'm going to actually use several examples from the field to really illustrate the magnitude of this problem. And the first example is from very close to my hometown in Los Alamos, New Mexico. And you can see this beautiful ponderosa pine forest that's really been hammered by fire, drought, and mountain pine beetle induced mortality. And actually Nate McDowell and colleagues did this really interesting in situ study of mature pinyon and juniper trees, where they exposed them to a 45% rainfall exclusion and a 4-degree warming. So end of the century climate conditions, arguably potentially more harsh than we'd expect with climate change because there's no CO2 fertilization. And this study actually occurred about a 20-minute drive from the picture I'm showing you. And what's really interesting is if you look at the study location on a map of the US where trees of the same species were dying of mountain pine beetle, it's smack dab in the middle of this mountain pine beetle driven mortality. But what's interesting is not a single one of the trees in Nate McDowell and colleagues' studies died from this climate driven stress in the midst of many trees of the same species dying due to pine beetle mortality. And one of the key factors was none of these trees in the study were infected. So the second example I want to use is from a little closer to my home today in Santa Barbara. It's from California's Sierra Nevada mountains during the 2012 to 2017 drought, which was a major drought in California. And my colleagues, Nate Stevenson and Adrian Doss from the USGS, have this really wonderful data set of several decades of detailed demographic measurements of plots along an elevation gradient in the Sierra Nevada. And what I show on the panel on the left is 3 of the main players in these plots: incense cedar, black oak, and ponderosa pine. And on the y-axis, I've plotted a trait that indicates xylem resistance to water stress, P50. And this is part of the parameters in that vulnerability curve that I talked about. And from this figure, you can see that incense cedar has potentially the most drought-resistant xylem, and ponderosa pine the least. And you could develop a hypothesis from a physiological perspective that you'd expect incense cedar to die the least on the landscape and ponderosa pine to die the most. But what's nice is we can actually look to see what happened because of this wonderful data Nate and Adrian have. And so that's what's shown here is tree mortality on the right 2 panels for each of the different species, which you can see along the bottom. And we actually broke it up for smaller trees and larger trees. This is tree mortality fraction. And if you look at the smaller trees, it's actually exactly the opposite of what you'd expect based on a physiologically based ordering. Incense cedar died at the most frequency, but actually going up to those trees, you can look and see that almost all of this mortality was caused by native bark beetles. If we look at the larger trees, it seems a little more physiologically intuitive based on this P50 values that we have for species averages. But going up to these trees, you can also see that almost all of the mortality is driven by native bark beetles. Mortality in both cases during this drought was driven by native bark beetles, and host selection differed strongly by size class, both within and among species. So what this really means is we could expect with some sort of insect disturbance interaction, a drastic underestimation of mortality based on physiology alone. And there's also potential for complete reordering of species depending on the host selection. And so I want to talk a little bit about why I think it's so important to build in a better process-based understanding of disturbance, including insect disturbance as well as other disturbances like drought and fire into these process-based models in the context of climate change. And that's really because forest disturbances are already increasing with climate change, so they're non-stationary, but we don't have a good understanding of exactly how much they'll change between 2020, which is where we are now, and 2100. And exactly how much they will change in this climate disturbance interaction is of really critical importance for understanding how the terrestrial carbon sink might evolve, and also understanding for the future of forest resilience and ecosystem services. And so just using fire as an example, because we have the most good data on this, if we look at using the MTBS database, what the 100-year integrated fire risk is across the landscape between 1984 and 2000, you can already see drastic increases across the Western US as indicated by the redder colors when you move to 2001 to the 2017 time period. And so we actually looked at using this MTBS database along with forest inventory and future climate data, how we might expect disturbances like fire, insect disturbance, and drought to change under different emission scenarios. And this is, we used a statistical based approach, so it doesn't include the moderating feedbacks of changes in forest cover, but it gives you a good idea of what the relative sensitivity is. And so we cross-validated these statistical models over the historical period, and you can see the historical is indicated in black here. And then we looked at changes in this case for burned area fraction across the US under lower emission scenarios, which correspond to about a 2.7 degree warming by the end of the century, which is notably higher than our target Paris Accord agreement, and higher emissions scenarios corresponding to over 4 degrees C warming. And there are a few features I want to point out for this burned area projections. First, you get, even though we've already seen drastic increases in fire in the West over the past several decades, it's really unprecedented and dwarfed by particularly changes in fire in the high emissions scenario. But actually these emission scenarios really matter, even if we're exceeding our target 1.5 to 2 degrees C. And particularly we get a pretty strong divergence around 2050. This shows very similar results for predicted drought mortality derived from forest inventory data along these different emissions pathways, as well as insect-related mortality. And the fire definitely shows the strongest trend, but this might also be due to data limitations associated with our insect and drought documented mortality that we rely mainly on the forest inventory for rather than these remote sensing products. So just to summarize for section 2, I showed that particularly based on Martin Venturis's Aspen Research Garden study, physiology is important in understanding drought mortality. But mechanistically predicting drought mortality at broad spatial scales using physiology alone has limited success, indicating that one or more key mechanisms are missing. And fire, insect, and drought disturbances are amplifying with climate change and interact with one another, resulting in complex mortality patterns across a landscape. And overall, I showed with Justin's work, broad-scale forest patterns at dry range edges can be anticipated from an understanding of plant hydraulics. Physiology is important in understanding plant responses to changes in water availability, but there's likely multiple missing mechanisms that are still important in anticipating observed forest mortality responses, some of which are becoming increasingly critical to understand with climate change. So with that, I'd like to thank you, and I'm happy to take any questions.
Great, thank you so much, Anna, for that wonderful talk. Folks can put questions into the Q&A, and I'll mediate the Q&A, but I thought I'd start it off and ask a question myself. Could you talk a little bit more about, in those future scenarios that you just showed, of the joint effects of fire and bark beetle and drought, whether those projections included interactions between the fire and bark beetles specifically? And if they didn't, how would you expect fire and bark beetles to interact as they both increase as disturbance agents moving through the next century?
Yeah, that's a really great question. It didn't include an interaction effect, and you'd actually, I think like water stress makes forests vulnerable to fire and bark beetle outbreaks because they're able to devote less carbon to defenses. The water stress declines live fuel moisture content, which is really fundamental in impacting wildfire spread. And so if you have these kind of compounding disturbances, you could maybe expect an overall moderation in total forested area, just because you can't necessarily both like burn a forest and have drought-driven mortality. But yeah, I didn't, and like what's documented as the ultimate cause of mortality is sort of like what we were relying on for those statistical models, and it didn't have any sort of compounding effects. But I think that's a really great question of like understanding their interaction in the future could be a really nice way to move forward, provided we have the data availability.
Great, thank you. So we have a question here specifically with the Sierra Nevada study where you had the longer term demographic data. Someone was wondering, was wondering, do you have an explanation for the lower than predicted drought mortality in black oak from that cedar oak pine study?
Yeah, so I think in all of the cases at the plots, like the predominant driver of mortality was this amplification of native bark beetles killing the trees. And so they weren't attacking the black oaks. And that's really why our physiological understanding kind of failed us in that case.
Great, thank you. So Gary Lovett asks, well, first compliments that it was a very interesting talk, and then asks, what are the physiological traits that can make drought-stressed trees more susceptible to insect attack? Are we able to assess and monitor those traits?
Yeah, so I think there's like part of the answer is we have some good ideas, and part of it is there's like a lot of unknowns associated with insect interactions and potentially novel pests and a lot of unknowns. But water stress trees are like, there's oftentimes a lot less carbon that's available to devote to insect defenses, which can really kick off some of these, if we're thinking about bark beetle events that predominantly occur during drought during the Western US, but then there's actually ends up being sort of a decoupling between stress and the ultimate bark beetle spread once an epidemic is kicked off. And so that makes it particularly difficult to model from a physiological perspective. And you might want to then employ some sort of epidemiological spread model to capture those dynamics after that kicks off. So there's some pretty complicated interactions that can initially be coupled to physiology and then get decoupled, which makes it a difficult problem, which is probably why we've had trouble making progress so far.
Great, thank you. This next question is from Charlie Canham and touches on what we've been talking about this morning, Anna. So given decades of ambiguous face experiment results, how confident are you about incorporating a CO2 fertilization effect in your model?
Yeah, so all of our current stomatal conductance models do incorporate like a CO2 fertilization effect. They've been parameterized based on they've been parameterized as best as we can based on the data that we've used, but they are empirical functions. We don't know exactly what the most appropriate data is to use. And, there are a number of different stomatal optimizations that can give you different results depending on exactly what sort of stressors the plants are experiencing. And so I think there's a lot of data behind like our understanding of increased water use efficiency and increased productivity with some CO2 fertilization, but really understanding exactly how, what the magnitude is and how strong of effect it's going to be is a major unknown, is what I would say. So I think that's a really great question. And, the face experiments were useful, but many of them were, limited in time extent and performed on not mature trees in a lot of cases. So I think there's a lot of opportunity to increase our understanding in that area.
Great, thanks. So from Peter Groffman, have you addressed the interactive effect of high temperature and drought, i.e. the hotter drought phenomenon that David Brashears talks about?
Yeah, so I guess in some ways we have, and in some ways we haven't. Certainly, all of these models incorporate the effect of temperature on vapor pressure deficit, and there are a number of other physiologically based functions related to respiration and VC max sensitivity to temperature. And, the plants in the model certainly experience like the effects of increased temperature. We resolve leaf level energy balances. And so if you decrease stomatal conductance, that certainly affects leaf temperature, but there's certainly also aspects of like these hotter drought conditions that probably aren't captured by our overly simplistic view of the system. So.
Great. So Kevin asks, climatologically, are we expecting more frequent droughts and more intense rainstorms? How could that affect tree physiology?
So it certainly depends on where you're looking across the globe, if what sort of extreme events you expect more of and like where you expect more precipitation. I think the general consensus is wetter areas will likely get wetter and drier areas will likely get drier. In California, we are expecting more frequent drought events. There's potentially like some accommodation, ecosystems will be able to accommodate some stress through things like intra-specific trait variation or species turnover, but I think ultimately if we are experiencing more severe drought conditions, that'll result in more frequent drought conditions, a net contraction of forested area in a number of locations. And that can also really exacerbate fire risk because live fuel moisture content, there's a number of like empirical thresholds that people have identified in the field as like making vegetation in different locations a really high risk for in terms of flammability. And we've already been seeing that play out across the landscape. I think sometimes even all the way over here on the East Coast, you guys have been blanketed in our secondhand smoke from the recent wildfires.
So Michelle Wong asks, first says, that was a great talk. And then asks, could you talk about the role of legacy effects in explaining species or regional differences in responses to drought?
Yeah, so I mean, you can imagine legacy effects occurring at very different scales. On the tree level, you could, and I talked a little bit about this, trees can sustain damage that only manifests in mortality after several years, because they're these dynamic organisms that can potentially repair their damage, but that comes at some sort of productivity and carbon cost. And ultimately the tree may or may not be successful in repairing that damage, leading to delayed mortality. You could imagine legacy effects due to different ecological interactions if you're measuring it from a satellite pixel. And looking at a 50 or 100 kilometer spatial scale. And so, I think differing legacy effects across the landscape are probably the product of these like organismal level processes, community level interactions, and also really how the subsurface is in a number of different ways moderating these effects, either through water availability, mycorrhizal associations, or other biogeochemical cycles. And that's something we really don't have a great handle on, just because it's some of these, particularly a lot of the subsurface processes are more poorly represented in some of our process-based models, and we just don't have a great idea of what's going on in the subsurface because it's fundamentally harder to sample than above ground, and our models are only a representation of what we know. So
Awesome. So from Evan Gore, and this may be our last question. First, very interesting talk. He says it seems that the biotic interactions are a major challenge, but not all species are similarly affected by outbreaks of biotic pests. So do you see better performance in upscaling physiological models for species that are less frequently attacked by pests?
Yeah, so that's a really great question. And I don't think, like for Martin Venturis's study, the biotic agents were driving all of that, all of those patterns. There are other, like Martin's study was performed for the Western US, and like subsurface water access is going to be a major driver of spatial heterogeneity in a mountainous region like the Western US. So, I'm not sure enough testing has been done to specifically show that, but I guess the bottom line is we haven't had great results at regional scale mortality prediction, even independent of like excluding species with bark beetle epidemics. But I do think a tractable way to kind of move forward and test that is to include, in model world, we have plant functional types, which are groups of species. You could imagine adding a few finite insect functional types and their interactive behavior, and sort of moving from there, from like a semi-empirical approach to a more process-based approach once we gain a better understanding and see some improvements. So.
Wonderful. Well, thank you so much, Anna, for taking the time to share your really interesting research with us. And Thanks to all of you out there for paying attention. I really appreciate it, and we will see you next week.
Thank you, everyone. And thanks, Wilson.